In this video, you shall learn about the various methods of artificial transformation in bacteria. There are two commonly used methods of artificial transformation. One is heat shock and the other is electroporation. The heat shock is the most commonly used method in the laboratory setup. So we shall describe this first. So let's begin with the heat shock method. In this method, we first take a solution of divalent cations, particularly calcium chloride. Why we use calcium chloride? Don't worry, I shall describe it in a moment. And we apply commercially available competent bacterial strain within the calcium chloride solution. Yes, nowadays we have competent bacterial strain that we can buy so that we know at least some of those bacteria are competent. And then we apply those specially prepared plasmids in the solution. These plasmids contain the antibiotic resistant gene and the gene of our choice, and also some associated regions, for example ORI site or region of replication initiation site and the promoter region, etc. We place this whole solution within ice at about 4 degrees Celsius for about 10 minutes. After 10 minutes, we transfer the solution in a warm water bath. This warm water is kept at about 42 degrees Celsius and we keep the solution there for about 45 to 50 seconds. And as you have noticed, this time is very short. This is because we don't want to kill those bacteria due to heat. So after 45 to 50 seconds, we immediately take the solution and transfer it back into ice. Same 4 degrees Celsius, and this time we keep it for 2 minutes. So as you have noticed, we alternate a cold followed by a warm and back into a cold cycle. During this cold heat cold cycle, the bacteria takes up the extracellular plasmids within them. Not all bacteria, some of those bacteria takes up the extracellular plasmid. So after 2 minutes, we transfer the solution in a shaking incubator. And we keep the solution at a temperature favorable for the growth of that particular bacterial strain, and we keep it there for about 30 minutes. We allow this time for those bacteria that have already taken up the plasmid to start synthesizing proteins from the plasmid, particularly the proteins that are synthesized from the marker gene, for example an antibiotic resistant gene. You will see the reason in a moment. Some laboratories use an optional centrifugation method. After this stage, we centrifuge the solution for about 5 minutes to concentrate those bacterial cells so that we can get better colonies when we plate from those concentrated bacterial cells. After that, we plate the bacteria in an LB agar medium containing that antibiotic, so that only the bacteria that have internalized the plasmids and contain the marker gene with the antibiotic resistant proteins within them that have already grown during the shaking incubator 30-minute period, only those bacteria will be able to grow in that medium. This kind of media is known as selective media. We keep those bacteria there for about 24 hours in a temperature that is favorable for the growth of that particular bacterial strain. Commonly 37 degrees Celsius is used in most laboratories. After 24 hours, we check if there is growth of any colonies, and if there is a colony, then we are sure that it contains the plasmid within them. Okay, but why do we use calcium chloride in the heat shock method? The bacterial cytoplasmic membrane contains a phosphatidyl glycerol structure. So it contains a glycerol molecule which attaches two molecules of fatty acid, and a phosphate group is attached to the glycerol molecule, which gives it a negative charge and makes it a hydrophilic region or polar region. This is the nonpolar segment or hydrophobic region of the phosphatidylglycerol molecule. Also, if you remember from the molecular structure of the DNA, that it contains a deoxyribose sugar which have a nitrogen base attached to it, and two molecules of deoxyribose sugar are bound to each other by a phosphate group. So this phosphoribose chain forms the backbone of the DNA. So as you have seen, both these molecules contain phosphate group which are negatively charged. So when the extracellular DNA material, for example the plasmid containing a DNA molecule, comes near the cell membrane, both of which being negative repel each other. When we apply a divalent cation in the solution, for example calcium chloride, it dissociates and forms calcium and chloride. This positively charged calcium ion, it neutralizes these two positively charged molecules and thus facilitates the entry of the extracellular DNA. So this is a widely accepted theory of why we use calcium chloride, but some scientists believe in some other theories too, and I shall briefly mention them. One of the theories is that the divalent calcium causes structural changes in the phosphatidylcholine cardiolipin bilayer that is present within the cell membrane of the bacteria. Also, some scientists believe that it induces phase transition in the phosphatidylglycerol within the bacterial cell membrane. You might also ask how the heat shock method itself works. So there are many theories too. One of the theories says that when we shift the temperature from 4 degree to 42 degrees Celsius, that is from the cold temperature to the hot temperature, it decreases the membrane fluidity and also releases lipid molecules from the membrane into the extracellular fluid. When we transfer the temperature back into 4 degrees Celsius, it increases the membrane fluidity and it releases protein molecules from the membrane. So use of these two temperatures alternatively can create pores within the membrane of the bacteria through which the extracellular DNA can enter within the bacterial cell. So this is one of the theory. Another theory states that the alternate use of hot and cold temperature reduces the membrane potential in the bacterial cell membrane. So as we know that the biological membranes have negative charge inside and positive charge outside, and as I have just discussed, the extracellular DNA molecules have negative charge in them due to the phosphate backbone. So although this negative and positive charge will attract each other and facilitate the transformation, but this intracellular negative charge will repel the the negative charge within the DNA and thus can inhibit the process of transformation. So by reducing this membrane potential within the bacterial cell in both sides, we can facilitate the entry of the DNA within the bacterial cell. So you might ask, well, we are using lots of potential stuff in all these heat shock methods, so can't we use something similar like electrical potential to create pores and make the bacteria transform? Well, yes, that's what we do in the electroporation method. So what we do in the electroporation? As the name suggests, it contains electro and poration. So we apply electrical charge or electrical potential in the media that creates pores within the bacterial membrane. So what we do is we provide short pulses of very low electrical potential, and this creates pores. But how does it happen? So as you know, the bacterial cell membrane contains a polar region and a non-polar region. The polar region contains the phosphate group along with the glycerol molecule, and the non-polar region contains two chains of fatty acid hanging from the glycerol molecules. So this is the structure of the phospholipid bilayer, with the polar region containing negative charges and the non-polar region without any net charge. So when we apply an electrical potential across these membranes, these phospholipid molecules, they reorient themselves and form a structure like this. So the polar region, they orient themselves in this way, and similarly in this part too. Well, you got the point. So as you can see, this creates a pore within the membrane through which extracellular molecules can enter within the bacterial cell, and these extracellular molecules might be those plasmids. So thus we can create bacterial cell membrane pore by using electrical potential, and this is what the electroporation method does. So what are the uses of the artificial method of transformation? We commonly use it to produce proteins, for example human insulin. We take the gene that produces that particular protein, we put it in a plasmid, and we put the plasmid within the bacterial cell by the process of artificial transformation, and we culture those bacteria and grow that protein commercially. Another use of artificial method is in the DNA cloning, where we can produce a large number of the DNA from a very minute amount of DNA by copying the DNA over and over again within the bacterial cell. And another use is in the DNA linkage studies, which I shall describe in a later video. So this was all about the artificial method of transformation. I hope you found this helpful. If you did, please like the video. Also don't forget to subscribe, because I publish new videos almost every week. Click the bell separate icon so that you get notified when I publish a new video. If you have any question on this topic, put it in the comment section down below, and I shall try to answer as many of them as possible. You can find useful links in the description section of this video. Also don't forget to share with your friends. So the next video will be on the bacterial conjugation. See you then.